Hi all, welcome back. Today I am going to give you a very simple lecture on the production of enzyme amylase. Amylases are a complex group of enzymes. They can hydrolyze polysaccharides like starch and glycogen to yield glucose. There are mainly three groups of amylases. They are alpha amylase, beta amylase and gamma amylase. Alpha amylase is a hydrolase enzyme. It catalyzes the hydrolysis of internal alpha 1,4 glycosidic linkages in starch. It is a calcium metalloenzyme, that is, it depends on the presence of a metal cofactor for its activity. Optimum pH is 7. Alpha amylase can be isolated from plants, animals, or microorganisms, and the enzyme is also present in barley and rice plants. Beta amylases is an exohydrolase enzyme. It acts from the non-reducing end of a polysaccharide chain. It can hydrolyze the alpha 1,4 glucan linkages to yield successive maltose units. The primary source of beta amylases are seeds of higher plants and sweet potato. Optimum pH of the enzyme ranges from 4 to 5.5 and it is used for the structural studies of starch and glycogen molecules produced by various methods. Gamma amylases, it can cleave alpha 1,6 glycosidic linkages and the last alpha 1,4 glycosidic linkages at the non-reducing end of amylose and amylopectin, yielding glucose. The gamma amylase, it has the most acidic optimum pH of all amylases because it is active around pH 3. There has been extensive research on microbial production of amylases. This is because of two major reasons. The microorganisms, they can grow rapidly and they will in turn speed up the production of enzymes. Microorganisms are easy to handle when compared to animals and plants, require lesser space and serve as more cost-effective sources. They can be easily manipulated using genetic engineering or other methods and can be subjected to strain improvement, mutation and other methods by which the production of the enzyme can be optimized. Different sources. The bacterial sources of thermostable alpha amylases are Bacillus subtilis, Bacillus stearothermophilus, Licheniformis, and amyloliquefaciens. Similarly, halophilic amylases can be obtained from Chromohalobacter species, Halobacillus species, etc. The temperature for the production is 45 degrees Celsius to 55 degrees Celsius, pH is 6 to 7, incubation period is 48 to 72 hours. Fungal sources. Mainly Aspergillus species and Penicillium species are capable of producing alpha amylases. Some examples are given below. Aspergillus oryzae niger, the temperature is 30 to 90 degrees Celsius, pH is 5 to 6, incubation period is 96 hours. In the case of Penicillium species, temperature is 30 to 55 degrees Celsius, pH 6 to 7, incubation time is 72 hours. Carbon source. Maltose, sucrose, glucose and oil cakes can be used as carbon sources. The nitrogen sources include both organic and inorganic sources. Inorganic sources are ammonium sulfate, ammonium chloride and ammonium hydrogen phosphate. Organic sources are peptone, yeast extract and soybean meal. The methods employed for the production of amylases on a commercial scale. Two methods are there, submerged fermentation method and solid state fermentation methods. First one is fungal amylase production. It utilizes the strains of Aspergillus oryzae and the method used is stationary semi-solid cultures. That is, we are utilizing this Aspergillus oryzae for stationary semi-solid cultures, and the Aspergillus niger can be utilized for submerged culture methods. In stationary semi-solid cultures, Aspergillus niger can be utilized for the production. The components of the medium are cornstarch, cornsteep liquor, potassium chloride, disodium hydrogen phosphate, calcium chloride, magnesium chloride, etc. And the problem of aeration and agitation is there because of the high viscosity of the medium due to the heavy mycelial growth. Stationary semi-solid cultures using wheat bran is employed extensively, and the organism employed is Aspergillus oryzae. Here, wheat bran is spread in thin layers in trays. Moisture it is provided by water and dilute acids. Sterilization must be done. Inoculation is done using spores. After the process, the medium and the fungus are dried at 50 degrees Celsius or a lesser temperature. Amylase can be extracted using water. The precipitation can be done by addition of alcohol. Finally, the product is dried at 55 degrees Celsius or less. Preparation of bacterial amylases. Bacteria employed are Bacillus subtilis and Bacillus diastaticus. High yield is obtained when Bacillus subtilis is grown in stationary cultures. pH of the medium is near neutral. Fermentation it proceeds for approximately 6 days. Temperature employed is 25 to 30 degrees Celsius. Bacillus subtilis it is forming a heavy pellicle growth and it is associated with the amylase yield. Fresh sterile air is circulated over the pellicle to improve the aeration. At the time of harvest, the culture is filtered and centrifuged. Concentrated by evaporation, salt and antiseptics are added. Amylase can also be precipitated by addition of ethanol, isopropanol or ammonium sulfate. Bacillus subtilis amylase can also be produced using submerged cultures employing a special highly starchy medium. The hydrolytic activity of alpha amylase can be determined based on the principle that starch and iodine react to form a blue-colored complex. On hydrolysis of starch, the complex changes to a reddish-brown colored substance. The absorbance can be read after the enzyme-substrate reaction has been terminated. This gives a measure of the extent of hydrolysis of starch by alpha amylase. Thus, we can test the hydrolytic activity of alpha amylase. Purification process. The methods employed are precipitation, chromatography, liquid-liquid extraction depending upon the properties of the enzyme desired. A combination of these methods are used in a series of steps to achieve high purity. The crude extracellular enzyme samples can be obtained from the fermented mass by filtration and centrifugation process. The crude enzyme can be precipitated and concentrated using ammonium sulfate precipitation or organic solvents. The precipitated sample is then subjected to dialysis against water or a buffer for further concentration, and this can be followed by any of the chromatographic techniques such as ion exchange, gel filtration and affinity chromatography for further separation and purification of the enzyme. This is the submerged fermentation method. Here we can see it is done in a huge production fermenter. After fermentation, the biomass is separated, concentrated, purified and modified. Then preservatives can be added. After that, drying and heating is done. Then the product will be either a liquid product or processed powder product will be obtained. Then in the case of solid state fermentation, here the fermentation it is done in tray fermenter, that is in trays or in rotating drum fermenters can be used. After solid state fermentation, this product obtained it can be concentrated, purified and modified. Then drying is done, then addition of preservatives, then the final product will be ready for marketing. Then next is the uses of amylases. Amylases are widely used in various industries. For example, they are used in starch processing, bakery and anti-salting. Then they are used in detergent and laundry industries. Then alcohol, textile, paper, food, pharmaceuticals, etc. These are some of the applications of amylase enzyme. This amylase it can be used for the starch conversion, that is for gelatinization, liquefaction and saccharification of starch. It is used in bakery industry for converting starch in dough to smaller fermentable sugars, used in detergent and laundry industry to degrade the residues of starchy foods such as potato, gravies, custard, chocolate, etc. into dextrins and other smaller oligosaccharides, used in textile industry, and it is used in fuel production, that is for converting starch into smaller fermentable sugars which are acted upon by yeast to produce ethanol. Then paper industry, It is also used for paper sizing. So these are some important applications, industrial applications of the amylase enzyme. That's all about the production of amylases. Hope you all understand. Thank you, and I'm wishing you all a very happy Christmas.